皆さん、こんにちは。LINK-Jの高橋です。本日は恒例となっている UCSD と LINK-J のジョイントウェビナーです。このジョイントウェビナーも今回で第13回目となりました。毎回 UCSD のインターナショナルイノベーションアウトリーチシニアダイレクターの和賀さんのご協力のもと、世界トップクラスの研究者による最先端の研究をご紹介いただく良い機会となっています。今回のテーマは、細胞内シグナル伝達と脂質研究、セッション3 with 神戸ユニバーシティということで、お伝えをしてまいります。脂質は生体膜成分やエネルギー源ということだけではなくて、脂質メディエーターとして重要な役割を果たしているということは皆さんもよくご存知だと思います。本日はどのような最新の研究を聞くことができるのかを大変楽しみにしています。本日はですね、画面でお示しをしているプログラムに沿って進めてまいります。ご質問はウェビナーの Q&A 機能からお願いをいたします。時間の都合上、すべての質問にお答えができかねる場合がございます。また、本イベントでは、ウェビナーの同時通訳機能、英語字幕をご利用いただけます。字幕は市販されているアプリを利用しておりますので、LINK-J UC サンディエゴ神戸大学は訳文の欠落など字幕に関する一切の責任は負いかねますので、どうぞご了承ください。また、本ウェビナーの録画、録音、または画面キャプチャーは固くお断りをしておりますので、ご了承ください。最後になりますけれども、ウェビナー終了時に表示されるリンクより、アンケートフォームにお進みいただき、皆さんのご意見、ご感想をお聞かせください。皆様の役に立つセミナーを開催したくご協力をお願いいたします。それでは、UC サンディエゴ国際イノベーションアウトリーチシニアダイレクター和賀さんよりイントロダクションとモデレーターのご紹介をお願いいたします。では、和賀さんお願いします。
はい、こんにちは。Hi, I'm Miwako Waga. I'm a Senior Director of International Innovation Outreach. My office has recently changed its name to the Office of Research and Innovation, reflecting the strategic importance of transferring research outcomes into innovations for the society. 2023 has been a very busy year for me in international partnerships. More people have visited the campus as if to catch up with the lost opportunities during the pandemic. I anticipate this trend to continue in 2024. There will be JP Morgan in San Francisco in January, Biocom Global Partnering Conference in San Diego in February, and Bio International Convention in San Diego in June. I look forward to welcoming many visitors from Japan at these events and other events in California next year. Now, let's ask Shirai Sensei of Kobe University to give welcome remarks about the joint webinar and invite Dr. Itay Budin. Thank you.
Hi. Very welcome to this session. I am Yasuhito Shirai, Kobe University. With Itay from UCSD, we have started this series of the webinar entitled Cell Signaling and Lipid Science. This is the third session. So we're gonna have two professors, Dr. Jin Chan from UCSD and Dr. Tomohiko Maihama from Kobe University, focusing on the signaling pathway in cancer. So both are experts of the cell signaling. I hope the audience will enjoy their talk. So Itay, can you introduce Dr. Jin Chan?
Yes. Thank you so much, Yasu, and thank you all for tuning in. As Yasu mentioned, this is our third of this fantastic series, and we have two great speakers today. The first is a dear colleague here at UC San Diego, Dr. Jin Zhang, who is a professor and vice chair of pharmacology here. Jin's lab is really the pioneer in developing new technologies for looking at cell signaling, especially using microscopy and fluorescence approaches, work that she has continued since she was a postdoctoral fellow here some years ago with Roger Chen. Jin has received numerous awards, really too many to list, but include ones from the Biophysical Society, the Protein Society, and she's also has been named an outstanding investigator by the National Cancer Institute for quite some years now. So with that, I would like to turn it over to Dr. Zhang and for her first talk today.
Thank you. Many thanks to Dr. Budin, Dr. Shirai, Miwako, and the LINK-J platform. Many thanks for this opportunity to present to this audience about our work. Let me get started. So looks okay? Okay, great. And so for the last 20 years, my lab has been interested in understanding how the cell organizes its biochemical activities to achieve very specific functions. At the molecular level, biochemical activities such as shown here, represented by kinase and phosphatase activities, are underlying every aspect of a cellular function. And these biochemical activities are also interconnected in a complex network like this. Each of these connections is made through protein-protein interactions, small molecule protein interactions, or enzymatic reactions. So these biochemical activities. And these type of complex networks are also embedded in the spatial temporal framework of a living cell, being constantly modulated. And that's why we've been interested in developing technologies to go into the live cell context to probe the properties, behaviors, regulation, and function of these macromolecules. And from our work and work from many labs in the field, we recently came up with this hypothesis. We hypothesized that cellular biochemical activities are not floating inside the cells. They're actually well organized. They're spatially organized into an activity architecture. We further hypothesized that disease states, such as cancer, reflect the reorganization and restructuring of this activity architecture. So we'd be interested in applying some of the technologies and molecular tools that we developed to probe this activity architecture across different scales, from the molecular scale to the nano micro scale to subcellular level and all the way to the whole organism level. And today I'm going to start by talking about some of the technologies that we developed over the years, and then present one example where we apply these technologies to learn something about this biochemical activity architecture. So shown here is a general design of our genetically encoded FRET-based kinase activity reporters. We call these CARs. We're building a lot of CARs in the lab. So as you can see, we're basically stitching quite a few components together and use that as a biosensor to sense kinase activities in living systems. Two of these components you may recognize are a pair of fluorescent proteins represented by cyan and yellow fluorescent protein here. Cyan and yellow fluorescent protein have the appropriate spectral overlap. So when they get really close together, they undergo FRET, or Foster resonance energy transfer. Of course, the efficiency of FRET is dependent on the distance and the relative orientation between the two fluorophores. So together they serve as the reporting unit of our biosensor. And we spend a lot of time engineering the middle portion, which we call the sensing unit. And for kinase activity reporters, the sensing unit consists of a substrate peptide that will be phosphorylated by the kinase of interest and a phosphoamino acid binding domain, which is a module that recognizes the phosphorylated form of the substrate peptide. So the idea is when the kinase is active, we'll phosphorylate the substrate peptide, and this phosphorylated form will fold into the binding pocket of the binding domain, thereby synthesizing a conformational change, bringing the two fluorophores close together, and we get a change in Fred. We and others have used this design to engineer a lot of cars, and today I'm going to talk about a story where we use eCar, which is for monitoring ERK kinase activities. And we have also used this type of design for looking at phosphatases. And this type of design can also be used for looking at second messengers. For example, cyclic AMP is a ubiquitous second messenger that's involved in regulating a lot of cellular processes. To monitor cyclic AMP dynamics in living systems, we developed these IQs or indicators for cyclic AMP using EPAC. We took one of the cyclic AMP effector molecules known as EPAC1 and sandwiched that between a cyan yellow Fred pair. And cyclic AMP binding to EPAC1 triggers a conformational change, which can be translated into a change in Fred that allows us to monitor intracellular free cyclic AMP levels and look at cyclic AMP gradients and nanodomains. And we have also used this type of design to look at lipid second messengers, since this series is focusing on lipid biology. So I think it's appropriate to talk about these lipid second messenger indicators. And for example, we're interested in looking at these 3 phosphatidylinositides, these lipid second messengers that are produced in response to, for example, growth factor stimulation. And we're particularly interested in looking at the subcellular pools of these lipid second messengers. To do that, we engineered Fred-based indicators that allow us to target them to different subcellular locations to look at subcellular pools of these lipid second messengers. However, when we try to engineer these lipid second messenger indicators, we encountered a challenge. We selected a plaque-gene homology domain, which has the right affinity and specificity to sense these lipid second messengers. But pH domain doesn't change its conformation that much upon binding to the ligand. So what we did is to engineer a pseudoligand, basically a stretch of acidic residues, to pre-associate with the same binding pocket of the pH domain. And when the real ligand comes in, it sort of kicks off or competes off this pseudoligand. And that engineered conformational change can be translated into a change in Fred, which allow us to monitor these lipid second messengers. Turns out that this design is pretty generalizable, so we use this design to look at quite a few different lipids. We can also engineer color variants that allow us to look at multiple species in the same living systems. And this type of design has also been used by Ryan Weeks, a recent PhD student in the lab, to engineer a RAS activity reporter. Here, we're using RAS binding domain to sense active RAS, which is the GTP bound form of RAS. And we also engineered a pseudoligand that pre-associate with the RBD. When the real ligand here, GTP bound RAS comes in, that will kick off the pseudoligand, which allow us to monitor RAS activity by a change in Fred. Ryan uses biosensor to look at the subcellular regulation of RAS, but also use it to characterize some of the RAS inhibitors, which represent an exciting direction in the lab. So these are Fred-based sensors. We have also engineered other fluorescent biosensors with enhanced capabilities. For example, these so-called X-ray series of sensors use single FP as the reporting unit instead of a Fred pair. And we achieved a hypersensitivity or ultra-sensitivity by using this design. And Soham pioneered this design in the lab. A current postdoc, Wei Lin, is our lab expert on these LINK biosensors, which we engineered to be compatible with super resolution imaging. So I won't go into the details of this design, just to mention this type of the sensors allow us to perform super resolution imaging to look at how kinase activity is organized at the nanodomain level. And Michelle Fry, a current postdoc in the lab, developed a chemigenetic sensors using Helotech, a self-labeling protein, to achieve both ultra-sensitivity and super resolution imaging compatibility. So these new chemigenetic sensors represented some exciting new directions in the lab as well. So I won't have time to go into details of some of the designs, and won't have time to talk about the other sensors, but we do have a biosensor database, which I listed here. We're in the process of updating the database, but feel free to check it out. Turns out we and others in the field have engineered many different sensors, and this database lists over 1,000 variants of different biosensors. And these biosensors are powerful tools to look at the spatial temporal regulation of cell signaling. Listed here are a few recent examples. For example, a PhD student, Shang Chen or Mingyuan Chen, used the phosphoinositide indicator to discover these lipid second messengers are not only present on the plasma membrane, they're also accumulating on the lysosomal membrane to locally regulate AKT, the important protein kinase. Danielle Schmidt, a former postdoc in the lab, started her own lab at UCLA, engineered an AMPK biosensor. This is a metabolic signaling molecule. And Danielle uses biosensor to look at lysosomal and mitochondrial regulation of this important signaling molecule. Jason Zhang, a former PhD student in the lab, also used this type of approach to discover one of the regulatory subunit of protein kinase A, R1 alpha, undergoes liquid-liquid phase separation to form these membranous organelles to enable highly specific cyclin P signaling. But today, in the remaining 30 minutes, I'm going to talk about one of our recent stories or recent discoveries where we looked at a GPCR-regulated ERK signaling. Shown here is the current textbook model of GPCR-regulated ERK. GPCRs, as many of you know, are probably the largest family of membrane receptors. It turns out that over a third of current drugs target one or many members or more members of GPCR family. And G protein coupled receptor, of course, turn on G protein, but many of them also activate ERK, which is a master regulator of cell proliferation and survival. According to the textbook model, beta-arrestin, an adapter protein that's involved in internalizing this receptor for the receptor desensitization, is critical for ERK activation. Again, according to the textbook model, beta-arrestin organized ERK activation machinery at the plasma membrane, leading to ERK activation. However, recently this model has been challenged. In fact, a few studies suggest it's the G protein rather than the beta-arrestin molecule that turn on ERK. So we wanted to really add the missing spatial information to this current picture to achieve a better understanding of GPCR-mediated ERK signaling. So this is what Yunhong Guang, a former postdoc in the lab, set out to ask. How does spatially compartmentalized GPCR signaling controls ERK? Yonghong just started his own lab at GIST in Korea. So when he was asking this question, he has a few different tools at his disposal to address this question. One set of tools are these e-cards, as I mentioned earlier. These are ERK activity reporters, genetically encoded, fluorescent-based. So same design as we talked about earlier, they're based on ERK substrate, which allow us to detect ERK activity, especially when we target the sensor to different subcellular locations by using these localization signals. So Yonghong took beta-2, a genergic receptor, which is the prototypic GPCR, and used a self-labeling protein labeled beta-2, and use SNAP dye to label the receptor so that we can follow the internalization of the receptor. And then just target the sensor, the ECAR sensor, to different subcellular locations, and use epinephrine, which is the beta-2 agonist, to stimulate the receptor and watch the ERK activity at different locations. So when we were first looking at the cytosol, we saw what we expected to see. Epinephrine stimulation of the beta-2 receptor stimulated a robust ERK activity in the cytosol, as expected. However, when we were looking at the plasma membrane, we got our first surprise. Although in the textbook model, it's been suggested ERK gets activated on the plasma membrane, we saw no plasma membrane ERK activity that we can detect. We know the sensor is functional because we can use growth factor to show the sensor is fully functional, can generate robust signal. So this experiment, together with some other control experiment, convinced us that we do not see, at least in the beta-2 case, any plasma membrane ERK activity. But when we were looking at the endosome using a endosomal targeted ECAR, and this sensor is targeted by using 5-domain, we saw robust ERK activity at the endosome. Again, this experiment together with some additional control experiment convinced us GPCR-mediated ERK activity, at least in the case of beta-2, occurs at the endosome rather than plasma membrane. Of course, the receptor gets to the endosome through beta-arrestin-mediated receptor endocytosis. So we next asked whether this ERK activity requires receptor endocytosis. So we block the endocytosis by two different ways. One is using barbitin to block this interaction. The other is simply knock down beta-arrestin. This is a critical player for regulating receptor endocytosis. So in both cases, as shown in yellow here, we lost the endosomal ERK activity stimulated by the GPCR. In the case of beta-arrestin knockdown, we can add back beta-arrestin 2 to fully recover the endosomal ERK activity. These data convinced us GPCR-mediated endosomal ERK activity requires receptor endocytosis. So what about the G-alpha protein? That's kind of the center of the debate. For example, what I was going to show, this is our textbook model, G protein on this side, beta-arrestin on this side to lead to ERK activation. But these are the papers that talk about the controversy. So we looked at this question by using a G-alpha as knockout line provided to us by the Innovate Lab in Japan. And in this line, as shown in gray here, we completely lost the endosomal ERK activity. And we can add back G-alpha-S to recover this activity, suggesting indeed this endosomal ERK activity requires G-alpha-S. When we were doing this experiment, we were watching the G-alpha-S. We can see it's initially localized to the plasma membrane. Upon stimulation, it comes out the plasma membrane and partially co-localized to the endosome. So this led to our hypothesis. We think the endosomal ERK activity requires active G-alpha-S at the endosome. And to test this idea, we used a nanobody known as nanobody 37 that stabilized the active open conformation of G-alpha-S And then we specifically target this nanobody to the endosome by using the same targeting motif that allows to specifically activate GRFS at the endosome. And this endosomally targeted MP37 robustly enhanced the endosomal ERK activity in contrast to the negative control, which is the nuclear targeted MP37 that shows no effect. We did a converse experiment using the peptide inhibitor for GRFS, also targeted to the endosome. And blocking GRFS at the endosome completely removed the endosomal ERK activity, in contrast to the nuclear control, which has no effect. We then looked at the receptor. We used yet another nanobody and targeted to the endosome. This nanobody will block the coupling between the receptor and G protein specifically on the endosome. And this also completely removed or abolished the endosomal ERK activity, in contrast to the negative control. So together, these data suggest GPCR-mediated endosomal ERK activity requires active GRFS and as well as the receptor at the endosome. What about the on the ERK side? We know ERK is activated by RAF1 and MEK1. This is MAP3K, MAP2K to activate ERK. By using inhibitors, we show both RAF1 and MEK1 are required for this endosomal ERK activity. We then looked at the association between GRFS and RAF1 and also GRFS and MEK1 by using proximity ligation assay. So the readout are these fluorescent puncta that will presence of these puncta indicate these two proteins are associated with each other. Indeed, in response to epinephrine stimulation, we can see GRFS and RAF1 are associated with each other. But more strikingly, if we stabilize the open active conformation of GRFS, specifically at the endosome, we got a 24-fold enhancement of the PLA signal, suggesting active GRFS at the endosome really enable the association between GRFS and RAF1. When we looked at a MEK1 and GRFS, we saw the same trend. This provides the evidence for a novel signaling complex consisting of GRFS, RAF1, and MEK1 at the endosome. Next, we check to see if this endosomally activated ERK activity will propagate into the nucleus, because ERK does a lot of things in the nucleus, including turning on target genes. We did exactly the same set of experiment. I won't go into great details. Instead of looking at the endosomal ERK activity, we use a nuclear targeted ECAR to look at nuclear ERK activity in the presence of these perturbation tools. So stabilizing the endosomal GRFS, enhancing nuclear ERK activity. Blocking GRFS on the endosome, block nuclear ERK activity. Blocking the coupling between the receptor and GRFS on the endosome also blocks nuclear ERK activity, suggesting activity could propagate into the nucleus. Also, all the nuclear ERK activity comes from the early endosome. We show this is not just for beta-2. For 3 other receptors, we saw the same thing. We then looked at the functional impact by looking at one of the ERK target genes, CMYC. CMYC transcription is upregulated in response to epinephrine stimulation, and that's dependent on this endosomal signaling complex, because we can block the GRFS at the endosome to block this increased transcription. And the same treatment, blocking GRFS at the endosome, also has a very strong effect on the cell proliferation, suggesting this endosomal GRFS RAF1 complex is very important for functional processes of the cell. Finally, look at the disease relevance by looking at one of the diseases known as MPS. This is a blood cell differentiation related disease, a form of a blood cancer. Jean Yao's lab, in collaboration with Roger Sunohara, who's also a collaborator on our project, has shown due to some splicing factor mutations, one splice variant of a GRS, this long form, is upregulated, and that mediates the disease phenotype in the ERK activity-dependent manner. We then looked at this by using the knockout line, and we express the long form and the short form. We look at the endosomal ERK activity and nuclear ERK activity. In both cases, only the disease-relevant long form is capable of inducing ERK activity. So together, we think these data start to revise the textbook picture of the GPCR-regulated ERK. We suggest there's a novel complex shown here. And we also think this has broad implications for biased signaling, which is an important concept that governs the development of GPCR drugs. And we that beta-arrestin and G-alpha-S collaborate in this case to turn on ERK. Finally, back to the activity architecture. I won't go into the details since I'm running out of time, but Soham and Jin Fan collaborated on doing these type of experiments in living mice, really allow us to look at the biochemical activity architectures in living animals. Finally, I think I mentioned all the main contributors and also collaborators. The in vivo work was done in collaboration with Rick. AMPK study was done in collaboration with Ruben. Roger's a key collaborator. Susan's a long-term collaborator on our PKA stuff. Our funding sources here. I'll stop. Sorry, I'm 1 minute over, but I hope to get some questions and to stimulate some discussion and collaboration in the future. Thanks.
Great. Thank you, Jin, for the this fantastic talk. I believe we have one question in the Q&A, so we can start with that. So it's a question from Takeara, asking if you'd have any thoughts on which kind of diseases can we potentially approach to develop treatments utilizing GPCRs or GPCR signaling, not only metabolic diseases, but ones that are cell-related, such as cancer?
Yeah, definitely. Thanks for the question, because I was running out of time at the end there, so I didn't quite talk about our perspective on the GPCRs in cancer. So actually, GPCRs have emerged, both GPCRs and also G proteins have emerged to be important targets in cancer. Actually, this is much of the development is thanks to my chair, Silvio Goodkin, who has been working on the role of GPCRs in cancer. So turns out a lot of these GPCRs and G proteins are dysregulated in cancer, and they can also be interesting targets for cancer immunology, in a sense for sort of manipulating the relationship between the immune cells and the cancer cells by manipulating the GPCRs and G proteins. So definitely the cell-related disease such as cancer could benefit from more GPCR drugs as well.
Thank you. See, we have a couple of minutes. Maybe I can ask the question. This role of GPCR signaling in endosomes is so fascinating. Do you think that there could be any factors that stimulate the segregation of the GPCR complex into an endosome, or is it more of a stochastic process that occurs?
Right. For the GPCR, for many of the GPCRs, such as beta-2, the GPCRs goes there through the beta-arrestin mediated receptor endocytosis. So traditionally, that's part of the desensitization process, because when we have the agonist on forever or for a long time, we don't want the system to be on continuously. So originally, this is kind of the part of the desensitization. So the receptor gets internalized. So presumably stop signaling, at least stop signaling from the plasma membrane. And that's kind of the part of the desensitization process for the system to deal with continuously on agonists. However, now work by Martin Losa, Mark Van Zestro, and now us to show the receptor, when it's internalized, they can signal to different things, still to G protein, for example, and also to ERK. So and quite often that's related to transcription-related events. So, but other GPCRs are not known to be internalized. And in that sense, in those GPCRs, it's an interesting question whether the internal signaling can also occur.
Great. And we have one a final short question asking from the chat, what is the role of ECAR4's EV?
So ECAR4's EV, so is this the, I guess are we, I wonder if this is a label on my slide. So if I'm not mistaken, we talk about the EV is EV linker. Is one of the long flexible linker that we incorporate between the substrate and binding domain that actually allow the FRET to be low in the basal state because they're as far apart as possible from each other. And this actually was pioneered by Miki Matsuda lab in Japan, where they engineered a whole bunch of EV sensors. And then we incorporated the EV linker into ECAR4 as well.
Okay. Thank you, Jen. And we can continue the discussion after the second speaker. So I'll hand it over now to Dr. Shirai to introduce Dr. Mahama.
Thank you, Jin Itai. I'm very pleased to introduce Professor Tomohiko Maehama. He is an associate professor of the Graduate School of Medicine of Kobe University. He has joined to our university in 2016 after many careers in the United States and Japan. His research interests are to elucidate molecular mechanisms of cancer development, particularly focusing on the function of tumor suppressors. So please present your talk, Tom.
Yeah, thank you, Shirai Sensei. Okay, let me share my presentation slides. All right, can you see the presentations?
Yes.
Okay, so all right, let me start my talk. So first of all, I'd like to thank Shirai Sensei and Dr. Budin Sensei for giving me an opportunity to talk about our study about YAP signaling and squamous cell carcinoma onset and progression. So I'd like to start my talk with a brief introduction of Hippo-YAP signaling system. Hippo signaling core is composed by 3 kinases, MSD and RATs and MDR kinases. SAB1 and MOB1 are the adapted proteins. When Hippo is activated by cell-cell contact, RATs kinase phosphorylates YAP and TAZ, leading to the exclusion of these proteins from the nucleus. In contrast, when Hippo is inactivated, YAP and TAZ go into the nucleus and activate gene transcription with T transcription factor, resulting in increased cell proliferation and tumor genesis. Okay. Since Hippo activation is induced by cell-cell contact, squamous epithelium in which cells tightly contact each other, could be largely affected by HIP signal disruption. So MOVAN depletion induces LATs and NDR activation, and eventually, MOVAN depletion induces LATs and NDR inactivation, and eventually activates intrinsic YAP and TAZ. Thus, we use MOVAN deficient mice to assess the effect of HIP disruption on squamous cell carcinoma genesis. We first generated MOVAN null mutant mice. Mice have two MOVAN paralogs, MOV1A and MOV1B. Thus, we knocked out these two paralogs and found that MOV1 complete depletion results in embryonic lethality. Detailed study shows that MOV1 is essential for post-implantation embryogenesis. In contrast, MOV1 partial depletion in mice results in spontaneous tumor onset in various tissues, such as skin cancer, exostosis, osteosarcoma, fibrosarcoma, breast cancer, and liver cancer. These tumors show up at 30 weeks of age, and all mice develop tumors at 70 weeks of age. From these observations, we decided to use conditional knockout system for MOVAN depletion in mice. First, we focused on head and neck squamous cell carcinoma, HNSCC. Oral cancer, including the tongue cancer, is the most common among the HNSCC. So head and neck region covered with squamous epithelium, so it would be expected that HIP disruption may provide a huge impact on head and neck carcinogenesis. On the other hand, P53 is known as the most frequently mutated gene in HNSCC. However, P53 inactivation alone never develops HNSCC in mouse model, suggesting other factors are required for head and neck SCC genesis. We hypothesized HIP-YAP signal plays a role in head and neck SCC genesis. Therefore, we explored the possibility using the MOVAN deficient mice. We generated tamoxifen-inducible MOVAN knockout mice using the Curie ERT system. Under this system, we directly applied tamoxifen onto the tongue of mouse in order to induce MOVAN depletion specific in the tongue. MOVAN gene disruption was observed at day 3, and MOVAN protein elimination was completed at day 7. We then tested tongue cancer development at 1 week, 2 weeks, and 4 weeks after the tamoxifen administration. In this mouse model system, carcinoma in situ showed up 1 week after the tamoxifen treatment. Then, all mice developed carcinoma in situ in 2 weeks after the tamoxifen treatment. And at 4 weeks, all tumor turned into invasive SCC. This result indicates the activation of endogenous YAP or TAZ is sufficient for such a rapid onset and progression of SCC. Next, in order to identify whether YAP or TAZ is involved in tumorigenesis, we induced additional knockout of YAP or TAZ over MOVAN knockout background. And we found that YAP depletion almost completely reversed the phenotype of MOVAN depletion, while TAZ depletion did not, suggesting that tongue cancer development is dependent on YAP. So next question is how YAP is activated in human HNSCC. We already know several genetic risk factors for HNSCC, such as EGFR, PS3 kinase, and P10, P53, and FAT1. Therefore, we tested the effect of these risk factors on YAP activation. P53 depletion and P10 depletion activates YAP by reducing the phosphorylation. FAT1 depletion and EGF activate YAP by increasing its amount. These YAP activations were also confirmed by nuclear translocation of YAP1. In addition, it is very surprising, we also found that these risk factors stepwisely increase the YAP activity. So P53 depletion slightly activates YAP. Additional P10 depletion further increase, wait a moment. Oh, I'm sorry. So additional P10 depletion further increase the YAP, and FAT1 depletion also additively increase the YAP activity. So NF2 depletion was used as a positive control for hippo disruption. I'm sorry, slide in this presentation is stacked. I'm sorry, wait for a while. All right, stepwise activation of YAP is also observed in human tongue cancers. So increased YAP expression was already found in the precancerous stage. And further YAP increase was found with the progression of human tongue cancer. In the second part of my talk, I would like to talk about hippo-YAP signaling in cervical SCC. Cervix is also covered by squamous epithelium, so we expected similar phenotype observed in the tongue cancer model. And the answer is yes, MOVA depletion in cervical epithelium result in early onset of CIS. We used the progesterone receptor query to induce MOVA disruption in the cervix. You can find early onset of cervical cancer within 1 week, and similar to tongue cancer model. So additional YAP1 depletion, YAP1 depletion reversed the phenotype of WOV1 deficient mice, while TAS depletion did not, indicating this cancer is also dependent on YAP1. So, however, we noticed one big difference from tongue cancer model. We found very slow progression of this cervical cancer. I'm sorry, wait for a while. Okay. So we tried to find out the reason for this slow progression. We first noticed that P53 was activated in MOVA depleted cervical tissues. We also observed MOVA depletion induced enlarged nuclei, aneuploidy, and micronuclei. Then we would be, this phenotype would be the reason for P53 activation. And P53 eventually repressed the cancer progression. In fact, additional disruption of P53 dramatically increased the incidence of invasive SCC. So it's times taken for the advanced cancer to develop was reduced from 40 weeks to 3 weeks. Risk factors for cervical cancer include HPV viral protein E6, E7, and estrogen, cigarette smoking, and several genetic alterations. We next tested these risk factors for YAP activation, as we did for head and neck SCC. We first focused on HPV E6 and E7. These viral proteins have several targets. We depleted these E6, E7 target genes and tested the effect on the YAP activation. We found that the depletion of PTPN14, as right here, which was an E7 target gene, significantly increased the YAP activity. Similar YAP activation was also observed in the normal cervical epithelial cells. So right panel shows E6, E7 expression, in fact, reduced PTPN14 protein, as like here, and activated the YAP in cervical epithelial cells. So, I'm sorry. So this is a reduction of the PTPN14 by E6, E7 expression and the activation of the YAP activity by E6, E7 expression. So in addition, estrogen and cigarette smoke extract activate the YAP as well as E6 and E7. PS3 kinase activation also activates the YAP. Finally, we noticed that these risk factors activate the YAP in a stepwise manner. Increase the YAP activity by estrogen or PS3 kinase, as like here. Okay. So increased YAP activity by estrogen or PS3 kinase was further increased PTPN14 depletion. So here is a proposed mechanism for SCC genesis. In head and neck SCC, YAP activity is stepwisely elevated by P53 mutation, smoking, PS3 kinase activation, FAT1 mutation, and EGFR activation. In cervical SCC, HPV plays a predominant role, and estrogen and smoking, the PS3 kinase additively activate the YAP. In both cases, when the YAP activity goes above oncogenic threshold, SCC develops, and further activation of YAP promotes SCC progression. So recently, we developed the breast cancer model by MOVAN depletion. I also would like to talk about this model today. So mammary tissues are composed of 2 layers of epithelium. One is luminal cell layer, another one is basal cell layer. Previous studies have demonstrated that invasive basal cell-like breast cancer, BLBC, are likely to have increased TAZ protein expression. However, the exact role of TAZ in BLBC remains unclear. We generated luminal cell-specific MOVAN deficient mice using the progesterone receptor Curie system. You can find PR Curie expression is specific for KRT8 positive luminal cells, in which Curie-mediated YFP expression was observed. In contrast, CRE-mediated YFP expression was not observed in the KLT5 positive basal cells. Under this system, luminal-specific MOVAN depletion result in breast cancer onset, as like here. So, as previously shown, the tongue and cervical cancers, MOVAN depletion in luminal cells result in a rapid onset of breast cancer. Ductal carcinoma in situ appeared within the 2 weeks after birth, then progressed to invasive carcinoma at 60 weeks of age. And metastasis to lung, lymph node, and liver was also observed at 60 weeks of age. TAZ has been suggested to be a potential driver for breast cancer. So we deleted TAZ over MOVAN knockout mice and found that TAZ depletion repressed MOVAN depletion-induced breast cancer development here, while YAP depletion did not. TAZ activation is also observed in the human BLBCs. TAZ expression was already found in very early hyperplasia stages, and the expression was elevated with the human BLBC progression. In addition, we found that these developed tumors were basal-like breast cancers, although MOVAN depletion was conducted in luminal-specific cells. These tumors have lost luminal cell markers, as here, but gained basal cell markers. When we tracked the time course of luminal and basal cell markers, we found that luminal markers have decreased during the cancer development. In contrast, basal markers have increased in the same specimen. We also found that ectopic expression of TAZ in luminal cancer cells induced luminal marker decrease and basal marker increase. Taking these together, we conclude that TAZ activation is a critical event for BLBC genesis. Although we don't know how TAZ is activated in breast cancer situations, several risk factors may participate in the TAZ activation. Then activated TAZ initiates breast cancer genesis by promoting luminal to basal transdifferentiation and tumorigenic transformation. In addition, tumors progress into invasive cancers when the other genetic alteration, such as p53 mutation, happens. So we have observed the YAP and TAZ play critical roles in tumorigenesis. These observations tell us the possibility that YAP and TAZ could be a candidate anti-cancer drug target. In order to search for potent YAP and TAZ inhibitors, we generated a reporter construct containing the YAP responsive element upstream of the luciferase genes. Cells harboring this construct show increased luciferase activity when YAP is activated. Using this YAP reporter system, we screened a chemical compound library. We divided first hit compound into 2 groups. Hit compound in the first group reduced the YAP protein, while hit compound in the second group did not. We finally obtained 3 potential YAP and TAZ inhibitors, allantractone and DMPCA as YAP protein reducing agent, and F04 as a YAP-mediated transcription inhibitor. Both allantractone and DMPCA effectively repressed tumor growth in vivo. Allantractone significantly repressed breast cancer cell growth in xenograft model. Allantractone was also effective on MOVAN-deficient tongue cancer development. DMPCA repressed bladder cancer cell growth in xenograft model. In addition, our brand new YAP inhibitor, F04, significantly inhibits YAP and TAZ-dependent transcriptional activity without affecting the YAP TAZ protein levels. F04 represses the breast cancer cell growth in xenograft model. F04 is very effective on breast cancer cell when compared to previously known hippo-YAP targeting anti-cancer agents such as beltfolfin, dasatinib, and MGH-CP1. This is a summary slide. We found MOBA deficiency in epithelial cell result in rapid onset of cancer, including tongue cancer, cervical cancer, and breast cancer. YAP activation, which would be induced by risk factors, plays a role in the onset and progression of head and neck SCC and cervical SCC. TAZ activation plays a role in the basal-like breast cancer onset. And finally, YAP TAZ may be a potential anti-cancer drug target. We are still working on the development of these types of drugs. This is a list of our collaborators. Thank you for your attention.
Thank you very much for a nice presentation, Dr. Maehama. So we got one question from the audience from Takara. Question is, according to this research, estrogen and smoking seems to be a factor for YAP1 and the following P53 cancer progression.
Yeah.
For medication, it would be great if you share both therapy and the lifestyle are possible to treat cancer.
It is a very difficult question because we don't know the exact mechanisms how estrogen activates YAP and reducing the P53 activates the P53. So there's a lot of several issues to be to answer to use this estrogen for the therapy.
Okay, thank you. So you show the three inhibitors. You found the three inhibitors for TAZ and YAP.
Yeah.
So those inhibitors can inhibit both TAZ and YAP because you showed TAZ is involved in the breast cancer, but not the YAP. But the allantogliptin, inhibited the breast cancer. That's why I thought.
That's a very good question because so we show just the YAP protein levels by allantogliptin, the DMPCA. So this YAP reducing agent, allantogliptin, the DMPCA, also reduce the TAZ protein levels. And this compound may work on both YAP and the TAZ. And another one is F04 is also effective on both YAP and the TAZ. So we don't show the results today, but we already confirmed the effect of the action of F04 on the YAP and the TAZ.
Okay, and one more I have a question. Let me confirm the activation of YAP. So you show the upregulation of the protein level of YAP in some cancers. And also you show the different phosphorylation level of the YAP. So which is important for the activation?
Yeah, that is very good questions because so the answer is both very important for the YAP activation. But unfortunately, we don't have a good anti-phospho-YAP antibody, which can be used for immunohistochemistry. So that's the reason why we presented just YAP protein levels in the human specimens.
So you showed a stepwise activation of YAP1. I thought there are multiple phosphorylation sites in YAP1. That's why they can be activated by
that's a possibility. So we are trying to find out the novel site of the phosphorylation or so previously known the other site of phosphorylation site. We are trying to find out which so draw a picture how so YAP activation is controlled by several upstream signals.
Okay, thank you very much. Itai and Jean, could you show up your face? Okay, so let's move to the final discussion and questions. So you guys have any question and opinion to this session?
So I'd like to ask Dr. Tsang. Is that okay?
Yeah.
Sure, of course.
So I'm very interested in the G alpha S induced activation of the ARC system. And so I know so people know the G alpha S may affect several signaling systems, such as the cyclic AMP production or something like that. So my question is, so for example, cyclic AMP doesn't affect the ARC activation.
We looked at that sort of by using a few pharmacological perturbations. It's actually the other way around. In this case, under our conditions, cyclic AMP-PKA play inhibitory role on ERK.
Mm-hmm.
So if we inhibit that pathway, we get even higher activity.
So this activations is just controlled by a protein-protein interaction. Is that right?
That's our model. Yeah, we
Mm-hmm.
think the G alpha S that's kind of recruited to the endosome will further recruit RAF1 and
Mm-hmm.
MEC1, then sort of organize the activation machinery that leads to the activation of ERK on the endosome.
All right, thank you very much.
Okay, I have a question also to your presentation, Jean.
Sure.
And you show the activation of ERK at the endosome, right?
Yes.
And so there are two types of the endosome, early endosome and the late endosome. So you detected the activation of ERK already at the early endosome?
Yeah, we sort of talk about it early endosome, but I think it's kind of leaning towards early endosome, but there are more specific, I guess supposedly more specific markers like RAP7, 9, 11, which allow us to differentiate that a little bit better. But right now, yeah, the data seem to hint that it's more of an early endosome, but we haven't really looked at it.
So if the ERK is already activated at early endosome, and RAF1 and MEC are recruited to the early endosome too?
Right. So we think that the RAF1 and MEC were recruited or are recruited by the activated or active G alpha S that leads to the ERK activation. So the kind of the sequence of event perhaps is more G alpha S, or of course receptor, and then G alpha S and RAF1 and then MEC1, and then that lead to the ERK activation. Although it's all happening in a relatively rapid fashion within minutes.
Okay. And one more, G alpha S is involved in many pathways, and you showed only case of the using the epinephrine. So in other cases, also ERK is activated at the endosome.
Right. Yeah. Actually, we are. That's a question that we're looking at now in the lab. So we show, in addition to beta adrenergic receptor, 3 other receptors behave similarly to beta 2. That's kind of the data that we included in the publication. But in the lab, we're also looking at additional GPCRs beyond the 4 receptors that we looked at. So yeah, it's actually interesting to start seeing some of the diversity. For example, we're looking at beta 1, which is you would think it's close to beta 2, but actually for beta 1, we because beta 1 doesn't get internalized, we do not see endosomal ERK activity. Yeah, instead we see
one more question from the audience.
Oh, yes.
Okay, the question is, the mechanism of how GRS-mediated ARC activation occurs, and only at the early endosome, but not the plasma membrane.
Yeah, great. I think I may have discussed with.
Yeah, you can.
Yeah.
You can see the question.
Yes, I can see the question, and I was gonna say I may have even discussed this with Itai, because we have two different models. Although we don't have experimental evidence, we think there are two things that are definitely different between early endosome and the plasma membrane. One is lipid composition, coming back to lipid biology. Lipid composition is different for these two membranes. Right. And the other is the curvature. Curvature is different for early endosomes versus plasma membrane. And both have been shown to influence the conformations of the receptors and G proteins, so especially the receptors. So that's where we think we need a certain curvature, we need certain lipid composition that stabilize certain conformation of the receptor that leads to the full activation of the pathway. So that's our current model, although we don't have evidence for experimental evidence for it yet.
Thank you very much. Itai, do you have any question and comment?
I mean, maybe just kind of an open-ended, we heard really, I think, two really fantastic areas of research of cell signaling and different approaches. And what do we think is potentially the future of combining some of the imaging modalities and tools that Dr. Zhang presented, and some of the more physiological models that Dr. Mahama presented, and do you think these kind of approaches can be combined in the future, and what are the limitations to that?
Yeah, for us, we're definitely very interested in going to in vivo, really perform using some of the further engineer the sensors to perform in vivo imaging to directly look at these biochemical processes in living animals. I kind of at the end very quickly presented one example, but we're definitely interested in doing more of that. Actually, for example, looking at cyclin MP, PKA activities in tumor slices, and also in vivo in the tumor of the animals, and also look at the sort of T cell cancer cell interactions in that say. Excuse me.
All right.
So yeah. Another thing is to use imaging for drug development. I was very interested in the inhibitor screening and try to think whether imaging can be used for that.
Dr. Mahama, can you see the question to you? Does YAP and the TAS have any important factors that induce cell competition?
So YAP is known to be involved in the cell competition process in some, limited conditions. YAP activation as the cells which has activated the YAP is so excluded from the normal cell layers. So it is a loser. It will be a loser of the cell competition. It is already known that. We published the papers a couple of years ago.
Okay, thank you very much. So, we are the kind of scientists who are interested in the pathway and especially the temporal or spatial activity of the enzyme. And as Jane showed, to see the activation of some enzymes at a very specific area at a specific point is very important. And I think it's a very powerful tool to develop a new type of medicine which can inhibit the enzyme at a specific area and at a specific time, maybe for the cancer treatment. But I think that was my hope, but for past 10 years. So I realized that to develop such kind of inhibitor is very difficult at this moment. Jane, do you have any idea to develop such kind of inhibitors?
Yeah, that's definitely our kind of thinking as well. Instead of inhibiting the entire pool of the enzyme, signaling enzymes, kind of using a sledgehammer, kind of fine-tune the activity and specifically activate or inhibit a subcellular or a selected pool of the signaling enzyme. But indeed, it's challenging, and that relies on better understanding of how these specific pools of enzymes are regulated. For example, I guess in some cases, a specific disease-relevant pool is targeted or linked to that location through a specific adapter protein, anchoring protein, then potential protein-protein interaction disruption type of inhibitors could be developed to kind of dislocate, right, remove that specific pool by using that approach. We have also encountered, I think there's another question, but I'll just finish. We have encountered another example, which is like one specific pool of the enzyme is modified differently by lipids. And then the lipid modification type of inhibitors could be used for targeting a specific pool.
Okay, thank you very much. It's our goal to do so. Okay, it's time to finish this session. Takahashi, please take over.
Okay. First of all, Dr. Chang and Dr. Maehara, thank you for sharing the cutting-edge research with us today. I would also like to thank Dr. Budin and Professor Shirai for moderating this session. Actually, it's on time. And Dr. Zhang introduced her research using a fluorescence imaging approach. She also proposed a new model regarding the GPCR-mediated ERK signaling pathway. Dr. Maehara introduced the HIPOYAP pathway is involved in the onset and the progression of the cancer, certain cancers, such as squamous cell carcinoma. He also showed the pathway could be the therapeutic target of cancer. So both presentations impressed the audience very much. And thank you again for sharing. Last but not least, I'd like to conclude this webinar session with my best wishes for further progress in research at both universities. Thank you very much for watching.
Bye-bye.
Thank you. Thank you for having us. Bye.
Thank you.
Thank you very much.